my name's Colin. I work at Microsoft Research in the biological computation group. So I'm going to talk about cells, the real biology cells, but I'm a software guy. So the only real difference is that rather than machine code that comes out, my compiler generates DNA. So other than that, it's not that different. Now, cells are often kind of referred to as like the building block of life, and they're kind of a natural unit in biology. And if we can work out how to program them effectively, as we do with digital computers, then there's a lot of breakthroughs we can make in terms of medical technology and clean energy and sustainable agriculture. It's also just kind of interesting hacking on living systems and kind of figuring out what they can do. So there's been quite a lot of talk in the industry about biologically inspired computing, so taking ideas from the biological world in terms of self-healing systems, genetic algorithms, and I think last year Jeff Hawkins was talking here, some of you may have seen, like computing like the brain. So what I'm going to be talking about is the opposite side of that process, so taking ideas from computing and putting them into biology, and how the kind of processes we use as software engineers can be helpful for the biologists as well. So some of the examples of the kind of things are like formal methods and state management, and there's a quite cool recent development this year from Drew Endy at Stanford University, where they developed what they refer to as the transcriptor, which is a direct analog from the electric transistor. So what you use it for is controlling the flow of biological computation, but in a biological setting. So I don't know if some of you might have been in the hardware hacking workshop yesterday. I mean, what our real vision is if we can download bits of biological code from the internet, drag them onto some kind of software environment, and then boot it up just like you're booting up an Arduino. That's what we'd like to be able to do. So there's three scales that we're interested in working on. So there's the cellular level, which is the natural area, and then below that there's molecules, how it's built up. And we also do some work on cell colonies. Depending on how many questions, I might skip the last section. So what we want to do in each of the cases, though, is have deliberate control. So design the system we want to build, simulate it, and be confident that when we go to the wet lab, it's probably going to work. Whereas at the moment, it's more like it's probably not going to work. So this is the kind of software we'd like to build, the kind of CAD, so kind of equivalent to the automotive and engineers generally have. So I'm just going to talk a little bit about what we've got to work with. So this is a DNA strand, and it's made up of a series of letters. So there's G, C, T, and A, and they go in pairs. So you always get a G and a C together, and you always get a T and an A together. They can be flipped around in both directions, so rather than binaries, 2 states, you have 4 different states. And it's not just the language that gets read like a programming code, it's also got definitions of the language itself. So you can kind of think of DNA in some ways a bit like a virtual machine image that you just add some water and then you're good to go. You don't need a kind of external definition of what's happening. So how do we work with this stuff? So you have the DNA codes or letters, and there's a special kind of protein called a polymerase, and this transforms a part of the DNA code into a strand, and then we transform this strand into what's called messenger RNA. So on the left, you can see the polymerase traveling along the DNA, and this does this about 20 base per second. So it's chugging along, doing the translation. After that, the messenger RNA is translated into a chain of amino acids by a special protein called a ribosome. Here we can see like the chain of amino acids coming out of the ribosome, and once the chain comes out, it folds into the protein structure. So what we've got is kind of a situation of proteins acting on DNA, producing more proteins that act on the other proteins. I hadn't really explained what a protein is. So this is a bigger kind of protein. So the protein is the one on the left, and it's quite a large one. It's an enzyme. And so it's got thousands of atoms. And the structure on the right-hand side is the substrate that it's working upon. Now, I'm not sure. I've heard from a lot of people like it's one of their ambitions when they're coming to Strangeloop to actually read Gödel, Bach, Escher. And there's a really good chapter in there, so if anybody has managed to get as far as chapter 16, you're kind of well prepared for the talk. And if you haven't, and you're confused at all, then you should go and read that to get some more clues. So, I mean, it's quite an old book, but a lot of the central ideas have kind of survived the test of time. And the only kind of things that happened in practice seem to be it's got more entangled. So there's more deeply nested, entangled hierarchies. So there's proteins also affect the DNA. So you've got this kind of circular situation where the proteins now affect the DNA, which produces the protein. And it's not clear what the origin of this process, but it's a self-booted loop. A bit like a compiler, you have to bootstrap it up and then keep progressing. So I'm going to talk first about molecular computing, so it's below the level of the cells. So as well as the other functions that the DNA performs, then it's also quite interesting as a chemical structure. So the letters themselves are made up of chemical compounds. And so we're going to go ahead and unravel the helix. So what you have now is two separated strands, and what's holding them together is fairly weak hydrogen bonds. So they go across the gap, and along the molecule you have, along the DNA strand, you have strong covalent bonds. So if you imagine a situation where you chop off a segment of the DNA, you can glue them back together in a lighter way. So the small ones have relatively few total hydrogen bonds. They form a kind of short helix, and it doesn't hold it together that strongly. So you can take those segments, get them together, and they'll fall apart. The longer ones will form a longer loop, and it'll be a stronger total bond. So that'll tend to bind irreversibly unless you specially intervene. So that's about all we actually need for being able to do DNA at the molecular level. There's lots of other approaches that need fairly expensive enzymes, but this approach is kind of sufficient for us. So I'm just going to actually now talk about some of the process that you can make use of it. So what we've got along the top is a strand of DNA, and along the bottom is the complementary section of DNA. So if those two strands were on their own, they'd bind very tightly. But this strand at the bottom has some bits bound onto it into the way. Now what we've got is what we refer to as toehold, which is the short red segment, which is an area where the DNA will, because it's short, it'll reversibly bind onto. So you can have situations where the molecules will naturally move back and forth. And just to clarify, obviously this is DNA. These things aren't conscious, they're not making decisions. It's something that happens as kind of stochastic, randomized process. So because it's made a short bond there, it's also able to push along the middle section that was tightly bound. So you can imagine a situation of it attaching to the DNA and then mushing along the other sections until it finally knocks off the other segment or strand of DNA. So what we've got a situation here is we've had one input, we pushed on another one, and something kind of different happened. Now because there's none of these toeholds or short sections left, this situation is now fairly stable. Now, that was a kind of arbitrary example. What I wanted to talk about is on the computing side, that you might be able to imagine a situation where you want to create a logic gate. So if you have some kind of cancer marker in a cell, it might be an indicator that there's some cancerous material out there, but things aren't always certain. And you want to be able to make deductions based on having multiple or sufficient quantities of cancer markers. So what we want to do is try and make an AND gate in this case. So we can again have the situation where the input will bind onto on the short segment and nudge off the section. And you can also imagine another situation where it'll bind on at the other end as well. So these sections in the middle can be pushed out, and then it'll displace the full output. So if you use this as a chain in the process of these inputs indicate something you want to detect in the cell, and that output will be acted upon by some other part of the system later on. Now, it's a bit of a hassle having to write out these DNA segments all the time, and you have to make sure they match, and so that's a kind of laborious and error-prone process. And so this is what computing is really good at. We give names to things, and then we're able to refer to them by those, and it's a more effective process for building systems. And so this is part of a language at the bottom that we've developed, and you can use it to program high-level behaviors and scale up higher. So I'm just going to give a bit of a demonstration of this process using the tool that we built. I was wondering, can anybody hazard a guess how you might be able to make an OR gate based on some kind of functionality like this? I can go ahead and show you if nobody wants to guess. Okay. Okay, I'll just make this big enough so you might be able to see. Okay, I'll just have to close the presentation. This is going to be harder. Okay, sorry for the delay. So what I've done here is I've defined up one part of the species. So this is going to be one input to the system, and then I'm going to define the other side. So what the syntax means in this case is the angle brackets are a strand that moves along the top. The numbers are the labels for sections of DNA. The carets are indicating that it's a toehold or short binding. So what I'm going to do to build an OR gate in the middle is, rather than having two pieces that need to be displaced, there's just one place that's shared between the two sides. Okay, so what we've got a system here is that the angle bracket, the curly braces are lower strands, and the star indicates that it's the complement, so it's the reverse of the DNA strand at the top. So we've got a kind of definition of an OR gate here. So what I'll do is now instantiate a certain size of the system. So what we're going to do is build a system with one of A and one of B and one of the OR gates. And now I'm going to go ahead and try compiling it, see if I've... Excellent. So what I've done is built a system, and this is showing you all the kind of intermediate states and intermediate species. And it can get quite complicated to understand these systems at larger scales. So we've built some visualization tools for indicating the state transitions. So what we want to do now is understand whether this system is actually going to behave the way we want it to in practice. So we've built a series of simulation tools for building these things on the computer and then running how they might behave. So this isn't going as far as molecular dynamics. It's a step beyond that. But it'll give us an indicator of whether these things are going to work in practice. So we can attempt to simulate this to see whether it will behave as we want to. And what we end up here is with a lot of the intermediate species, which kind of gets in the way of seeing what's going on. So here what I'm going through is the process of just indicating these are the only species that we want to see in the output. And so what we've got is a reducing amount of species A and a reducing amount of species B. So what we've done in this case is what's referred to as a deterministic solution. We've taken the maths and done a calculation as if these things can be infinitely divided. So in the real biological situation, we don't have things that can be broken apart. These are like our minimum unit sizes. And so another way of analyzing the situation is a stochastic simulation, which in that situation you imagine these bits and they interact and they transform, but you can't divide them up. And so if I try that, sometimes what will happen is one of them will be transformed and none of the other one. So these things become a lot clearer if we have a few more of each. So what we've got here is some of the inputs being transformed, and that sometimes they'll bind and unbind and then go back to floating in the super stuff, but you are getting transformations into the output. So we could equally do this kind of simulation with the AND gate, and that would then only work if the were quantities are each to be used up. Okay. So what I showed you there was a fairly toy example. And so with lots of the collaborators we have with experimentalists as well as theory people, they've taken this approach, and sometimes with that tool itself, and built up chains of these logic gates. In the same way kind of an electronics engineer would use multiple logic gates in order to provide a function, you can also chain up these sets of behaviors as well. So one of the more concrete things that we've done with this is there's an idea in biology called a DNA aptamer. This is like a little chain that you can make, and you can take it between something that an organism already knows about and something that's kind of, it's naive to, it hasn't been exposed to it. So an experiment was done where they took a pathogen, in this case anthrax, and some mice, and so there's some pathogens that the mice were already exposed to. And if you build an aptamer that directly binds between anthrax and this existing known pathogen, then what you create is a kind of instant immunity. So the mice could be given as much anthrax as you wanted to, and they weren't affected at all. So it wasn't so good for the control case. But so this is another more kind of natural to the biology sphere. So there's this kind of ongoing debate that a lot of the engineers who are involved in biology now, they want to make everything into a logic gate, but there's also a more direct way in some respects of directly building the analog decision-making process. So what we wanted to do was build an algorithm that would judge the majority that was existing in a cell. And so you could tell whether there's more of the bad stuff or more of the good stuff, and then make a determination of whether you want to do something about that. So what we did was build a kind of series of chemical reactions. So this is kind of an indirect way of going about it. Where you have to do it in practice is even more indirect. But what you're doing is every time an X or Y meet, they all combine and turn into some kind of intermediate molecule. Every time an X bumps into this intermediate molecule, it turns into 2X. And every time a Y bumps into an intermediate molecule, it also similarly turns into 2Y. So the reason this in practice figures out which is in the highest concentration is the probability of an X bumping into an intermediate molecule is proportional to the number of X's that already exist in the system. So the more X's there are, the faster it will move in that direction. So just to kind of give you a little bit of insight into the process of building more complicated systems, then these are the strands and processes that we have to put together to make it fast enough in a biological system and also reliable. And so as well as wanting to make these kind of decisions, there's also times when we want to perform more numerical operations. So things like counting you can do fairly well in cells and make decisions on that. But people also want to do things like calculate square roots. And so this fairly long series of strands, which you can probably make out somewhat, is one implementation of a square root circuit implemented in molecular computing. So the kind of question is, is this correct? And so we have good techniques for this in computing that you can use a kind of formal methods approach. So probably a lot of you might have used like SAT solvers and the like. And so one of my colleagues and collaborators have worked on applying the Z3 SMT Solver to the biological case. And so I'm not going to tell you whether this one works because they're going to be announcing probably next week at a DNA conference. But I can say that some published DNA implementations don't work very well, and some work better. Okay, so doing the time. Okay. So I said I was going to talk about cells, so we're going to move up to that level now. And one people always kind of obsess about in computing is like how powerful your computer is compared to all the other computers. And who has the most megahertz and the like. I mean, in lots of ways, then cells put that current computers to shame. They're not so good on the megahertz, but they are massively parallel. They're robust to failure. They build themselves, so you don't even have to buy them yourself. And they're also very efficient in terms of the calculations they perform for the amount of energy that you provide them with. So one of the other nice features is that they're actually, in some instances, quite cross-platform. You can take the DNA out of the jellyfish and transplant it, move it into a bacteria, and it still glows. So the function that represented the glowing capacity in jellyfish is general enough that it can work across species. Now, there's lots of things that don't work this way, and it's hard to translate them across. So you're not going to be able to get DNA and just glow for parties or anything like that. Well, you probably wouldn't want to risk it either. But now, in terms of the efficiency of storage, then there's this quite interesting paper where the blue ones are basically our current technologies. These are the things that the electronics have been able to make. And the purple color is the kind of the borderline physics approach. And so on the x-axis, we're kind of indicating how much we've been able to encode into this system. So things off to the far right, we can put a lot of information there. And to the far left, we can put very few things. And the Y scale is how dense it is. So how big the thing is you have to produce in order to have that amount of information. So there's some quite interesting work of encoding the information you have to store into those letters of the DNA. And so they're quite good in terms of amounts of density. So you can fit it into a very small area, but they're not really up there yet with the total amount. It's also a bit expensive at the moment, so it's about £10,000, $10,000 per megabyte to write, and $200 to read. So that's not really going to be very appealing to anybody just yet. But one of the very interesting things, there's some quite interesting trends in economics of DNA reading and writing that, in general, then electronics will move up about a factor of 1.6 per year. So you'll get that kind of gradual increase, whereas the historical trends on DNA reading and writing are going up at much faster rates, between 5 and 10-fold at times. And so this is kind of one of the areas I was interested in getting when I started in the field, that like almost nothing moves faster than Moore's law in terms of software development areas. And so any field where there's trends where the basic cost of doing things is going down at a greater rate than Moore's law, you can kind of see some disruptive potential. So if you could make something work now, just about, then in a couple of years, you might be onto a very effective situation. So there was kind of suggestions, maybe 5, 10 years, it might be commercially viable to be storing long-term data storage in DNA. So there's also another way of looking at it, being able to write DNA, that the bigger systems we can build for a cost-effective manner, then we can build more interesting systems. So this is a guy called Rob Carlson that he's made kind of an analogy between two quite different things. There's debate whether it even makes sense. So what we're measuring is the number of transistors per chip in blue, this is what Intel and AMD are doing. And in orange and red, that's reading the DNA. And it's measured in a slightly odd measure of bases per person per day. So it's a person physically doing that. And in yellow, it's writing the DNA in a similar basis per person per day. So there's one company that's kind of going for this process called Amaris. And so what they've built is a kind of synthetic biology route to producing one of the key ingredients that goes into malaria treatment. So they've got this up and running, that rather than producing artemisal acid through an agricultural route, which is quite difficult and expensive, then they have a biology reactor type system where they've taken DNA, gone through a design process, and put it into the cells. Now the problem is they had to go through a lot of engineering and research to get this far. And so something they've kind of publicly said, and they work on themselves, is they themselves want a kind of software process for being able to do this. So there's expectation, and Jay Kiesling, one of the co-founders of Amaris, said that like in almost all areas of engineering, then you have simulation tools. The idea that you just build something and find out if it works isn't really very effective compared to being able to follow this rational process. Okay, so we've still got all these problems, like we're kind of hacking into these systems because we didn't write the original code a lot of the time. We're trying to find pieces that we want to make use of. And there's all kinds of challenges that they sound a lot to me like the software engineer's kind of experience as well. That you haven't got the present system working properly, but you're still expected to add more functionality on top of it. And so having some tools that can help you out with that seems like it would be quite nice. So this is one of the things we've been doing, that it's a piece of software for expressing the behavior of the system and then getting the process out. So I'll just play a little bit of the video, which is showing the DNA transcription process. So this is the polymerase attaching, and then it's going to move down the DNA chain and produce the messenger RNA. So it's quite a fun video, but I'll move on. So this is the current process. We've got a low-level view of DNA that we have the start of the DNA where we want to bind onto, which is called promoter, and we have the end, which is a terminator. So these are sections of the DNA that will move along and do. So there's kind of interesting things that there's overlaps, and this is its way of communicating. And so the polymerase needs to move along this system. And so what we've had to do in each of these cases is provide the very specific elements for these parts. And it's not helpful, because if you move one part out, then you can cause the rest not to work. And this is, in a sense, the hello world of producing something. That this isn't what we want to do. We want to produce far more significant systems. And if this is hard and unpredictable, and making minor changes causes unexpected outcomes, we haven't got really a hope of producing bigger systems. So what we want to do is say, this is what I want you to do, fill in the parts for me. So what I'm going to do is another kind of toy example, which this is one of the very early things in synthetic biology called the Repressilator. And what we've got here is we've got compound A and B and C. At least that's what I'm going to say rather than the biology versions of it. That what happens is that A inhibits B and B inhibits C, and then C inhibits A. So you've got a system where if you've got a lot of A, then that'll suppress B, which means C is going to go unchecked. And C will then increase, and that will start suppressing A. So you've got a dynamical system in practice. So another tool. So what I'm going to do is skip ahead to the example rather than typing it out this time. So this is our expression of the system. So we have a coding for A, and we have a promotion against the inhibition of C. So these are the three different parts of the system, and we've also got a database of genetic parts. And so what we're going to ask the system to do is now solve this for us. And it's produced quite a lot of solutions. As you can kind of see from the comment, then one of them's good and one of them's not so good. So I'll kind of show you to start off with why the first one isn't so good. And the way we do this testing in this case is, again, with simulation. So what we've got here is just one part of the system that's going kind of out of control. There's no oscillations. There's no the other parts coming into play. So what's happened here is the genetic parts aren't really compatible for the function that we've described. So rather than going to the lab and finding this out, you can find this out to an approximation in silicon. So instead, if we pick the better solution and run the simulation, and this is always risky because it's a randomized process, so it could just not work, but that would be very unlikely. But we've got peaks here of the green and then the red and the blue. And it's not perfect. There's like a lot of noise in the system, which is a fundamental part of it. But it's what you deal with in a biological system. So you'd also need reinforcement and things like the approximate majority circuits built on top of this in order to get reliable behavior out of the end. Okay. Okay, and in a kind of similar way that I kind of showed you a toy example, but these tools are actually used for real research purposes. This is a model of a part of the immune system that when a cell in your body gets infected, then the cells themselves the cells communicate what's inside them because your immune system wants to come around and inspect what's happening. And so the mechanism for this happening is the MHC class 1 system. So it takes fragments of the viruses that might have got inside your cells or parts of the cells themselves and moves them out to the outside. So this is a quite involved mechanism, but using this same tool which we used for the other example, then we're able to explore how variables act on this. We're able to collect experimental data and then train a model that was better able to predict real behavior. And so then some of my colleagues have published work based on this. Okay. So the choice is, if you've got questions, I can answer questions, or I can talk about cell colonies. So I guess an indication, are there lots of burning questions? Yeah.
So your time axis, how are you simulating time there? Is it actually based on the chemical dynamics?
Yes, it is.
Stepwise, discrete, stepping.
Yeah. So the x-axes are real time periods. Most of the inputs for this kind of system, they're not set up for real work. So they're not, the parameters that are fed in aren't accurate. So therefore, the time periods won't be true. If you go and collect characterization data and then use machine learning techniques to train the models, then it would be accurate. So I think I'll keep talking then, if we don't have any questions.
Quick thing, for your DNA circuits, what are you assuming for the stabilities of the different intermediates? Because your delta G values for your products are only marginally stable. The back reactions are going to occur. And I assume you're using that in your stochastic simulations as well. So what underlying thermodynamic models are you using for that?
So I'm not familiar with the term delta G. We have, okay, so we have,
Determine the equilibrium constants of the reaction.
okay. So the reactions will go back and forth. So the short sections will bind and unbind. I mean, in terms of, I don't know if you're asking questions about molecular dynamics or more deep than that.
Well, you have an OR gate,
Yep.
and the products are very stable. You're going to have an equilibrium constant, right? And if that reaction isn't stable, you're going to go and do back reaction as well. So in your model, are you assuming 100% completion of the reaction? So you have only products left to go on to the next stage of the reaction?
No, you wouldn't necessarily have a full transformation, and it's going to asymptotically approach the final state. So this is partly why we have a lot more involved solutions for real-world problems, that it won't go close enough, and so you'd have to build more circuitry on top. So a lot of these biological systems, they don't need to reach a full final state in order to act. You have the things you're trying to achieve, for example, treating medical conditions, you only need to treat a certain number of the cells of itself anyway. Question there?
What is the mechanism for actually translating a given string in the computer ABCG into physical form?
There's some quite helpful companies that you email them your DNA that you want, and they'll fabricate it for you. So, slightly distressingly, I think they took inspiration from Web 2.0. So there's a company called DNA 2.0 that. So there are certain restrictions, as you might imagine, that people might have the DNA for something that's somewhat harmful, and you don't necessarily want people to be able to order off dangerous biological material. But in general, as long as what you ask for isn't expected to be dangerous, they'll send you biological samples from your specification. Any more? Okay. So I'm just going to talk briefly about the cells moving together. So what we have is things like biofilms, which are colonies of cells, which there's a lot of different types of cells, but together they form a hard to undermine colony and resistant to antibiotics. So what we've done is on GPUs, and I can't give a demo of this because this laptop doesn't really have a good enough GPU in it, that we're able to predict how these cells will interact and carry out experiments. And one of these is to take a prediction of how much of the interaction between cells is caused by their physical shape. So what we've done here is taken out the situation of rod-shaped shells, so elongated cells, or just round cells. And we've stained the different colonies, so the original cells that made up these parts, and then grew them on the computer. And so purely from the physics of the situation, then there's been penetration into these different areas. So I think that's just about it. So I'm just going to wrap up now. So what I've shown was both the DNA computing and the molecular computing area, and also the kind of forward engineering of cells in a term called synthetic biology. Some of the other things that the group I work in does is trying to understand rather than engineer systems. So how the immune system behaves and how cells develop. And so underlying all of this, we have a kind of shared library, and that has a lot of functions for the simulation and the analysis of the system. And we can also output and produce solutions that are based on this using external code as well. And so this is the group I work with. So there's a lot of researchers, and I've shamelessly stolen lots of their work and put it in here, so I'm very grateful to them. And I'll just leave you with some of the other things that we're kind of working on.